Okay, so hello everyone, and thanks for joining us in this edition of the RNA Collaborative Seminar Series. This time is brought to you by the Riboclub in Canada. For those of you who do not know the Riboclub is really a network that groups all RNA researchers in Canada, and it covers a broad range of RNA topics. And every once in a while, we actually select for you two speakers to share with you and present to you what's actually been we're doing in Canada in terms of RNA science. And today, the two speakers are actually in one level are very, very different, discussing very different topic, and another level actually have some kind of similarity between them in terms of, as you would see very soon, they actually sharing kind of interest in developing tool and actually bringing service to RNA community beside the kind of our usual RNA discovery and research. And I'm hoping that today you would find that those two seminars are really very interesting, both on a topical level, but also in terms of tool development and utility for your own research. We'll start, and if you don't know me, my name is Sherif Aboulela. I'm a professor at the University of Sherbrooke, and I'm the coordinator of the Riboclub. Our first speaker today is Michelle Scott from University of Sherbrooke. Michelle actually did her PhD in McGill at McGill University with a professor of David Thomas and Michael Hallett. And she actually, her background was in kind of biochemistry, molecular biology, and computer engineering. And during her PhD, which finished in 2005, she worked mostly on predicting protein subcellular localization. Later on, she moved on to University of Dundee in Scotland to finish her postdoctoral training in 2011 under the supervision of Dr. Geoff Bartons. And she actually progressively migrated at this point towards interest in RNA research. She started by studying protein localization in the nucleus, and then probably she found the nucleolus, in the nucleolus, and definitely by looking at the nucleolus, she got increasingly interested in the RNA that found in the nucleolus and started working a lot on small nuclear RNA. She joined the University of Sherbrooke at the Department of Biochemistry and Functional Genomics in 2011, where she's currently a full professor. Her group's working primarily on snoRNA biology and transcriptomic using computational approaches, including machine learning. Her research is supported by many different like funding agency in Canada, including the CHR, which is the equivalent of the NIH in the States. She's also an FRQS senior research fellow, has an FRQS research fellowship, which is a prestigious fellowship in Québec. She developed many different tools. She also hosting many different databases. And today she was talking about the link between small nuclear RNA, ribosome biogenesis, and transcriptome-wide analysis of non-coding RNA, especially small nuclear RNA. With that, I will stop here, and I will invite Michelle to start her presentation.
Thank you very much, Sherif, for this very nice introduction. And I'd like to thank the organizers in general for inviting me to participate in this far-reaching seminar series. And today I'll tell you about our recent work studying RNA interactors of small nucleolar RNAs, or snoRNAs. So snoRNAs are an ancient class of non-coding RNAs. They're present in all eukaryotes, and they've been best characterized for their role in ribosome biogenesis. So many of them have been shown to act as guides for the site-specific modification of ribosomal RNA. There are two main classes of snoRNAs, the box CD snoRNAs and the box HAC snoRNAs, that differ in terms of their sequence motifs, their general structure, the proteins they interact with, and the chemical modification that they catalyze. But in both cases, they'll identify their substrates by directly binding to them, so forming an RNA duplex with their substrates to specify the exact position that requires modification. In human, there are over 2,000 snoRNAs that are annotated in genomic references. But only, well, fewer than 600 are annotated as targeting ribosomal RNA, and fewer than 200 as targeting sNRNAs, which are the canonical substrates known for snoRNAs. And there's overlap between these two groups. So overall, fewer or less than a third of the human snoRNome is actually predicted to be involved in these functions. And I can add that together, these snoRNAs are responsible for the modification of 210 positions in ribosomal RNA and 34 in sNRNA. And given that one snoRNA can modify more than one or two positions in these substrates, we can conclude that there's redundancy in the system, so several snoRNAs can modify the same position in the substrates. So what might the remaining 2/3 of human snoRNAs be doing in cells? Well, over the past 15 years, there have been many reports of snoRNAs involved in other functions in cells, mainly in the regulation of gene expression. So some snoRNAs have been shown to be able to guide the modification of other RNAs, like, for example, messenger RNAs. And in one case, this has been shown to be able to regulate the translation of this messenger RNA. Other snoRNAs are involved in protein trapping. So some of them will sequester splicing factors, resulting in changes in the splicing landscape. And other snoRNAs are involved in sequestering 3' end processing factors, which will result in changes in the choice of the poly A tail position for a subset of messenger RNAs. Other snoRNAs are involved in the regulation of alternative splicing by directly binding splicing regulatory elements like splice junctions, and presumably competing for binding on these elements with the spliceosome. Then there's other snoRNAs that are enriched in chromatin and believed to be involved in chromatin remodeling, so changing the accessibility or the opening up of chromatin. And then there's one snoRNA that has been shown to be further processed into a piRNA that can recruit the nuclear exosome to specific pre-messenger RNAs, resulting in their degradation, so affecting their stability. So overall, there's now many examples of snoRNAs that are involved in these non-canonical functions, but in all cases, only a very small number, one or a handful of snoRNAs have been experimentally validated as carrying out these functions. So we really don't know the extent of the involvement of snoRNAs in these functions. And we also don't know the relationship between the role of snoRNAs in ribosome biogenesis and in these non-canonical functions. Some snoRNAs are involved in both, but we don't know the relationship between these functions. And I can add that for these non-canonical functions, almost all of them have been described for boxed-CD snoRNAs and not boxed-HTC snoRNAs. So for now, most non-canonical functions have been described for boxed-CD snoRNAs. So my group is interested in investigating how common are ribosomal RNA and sNRNA independent functions for snoRNAs. And one of the members of my group who was most involved in this project is Gabrielle Deschamps-Francaire, who just defended her PhD last Friday. And she spent several years of her PhD laying the foundation so that we could start asking these questions. And most of what I'll tell you about today was very recently accepted for publication in NAR. So the first questions that Gabrielle started asking, or the first things that she started doing on this project, is obtaining the targets of all snoRNAs so that she can compare them. And she was working on boxed-CD snoRNAs because those are the snoRNAs that are most characterized as involved in non-canonical functions. So she started by obtaining the list of all the targets of boxed-CD snoRNAs, all the canonical targets of boxed-CD snoRNAs in ribosomal RNA and sNRNA. This is very easy to do because there are good databases that provide this information. She also curated the literature to obtain the information about all snoRNAs that have been shown or experimentally validated as carrying out non-canonical functions, regulating gene expression of specific, mostly messenger RNAs. And that's what we're looking at here. And when she compared these two sets, so in the case of the canonical targets of snoRNAs, all of them involve binding to what is known as the guide region in box-D snoRNAs. So this is a region that's found immediately upstream of the box D and or the box D prime. But in the case of the non-canonical functions of snoRNAs, they can involve any portion of the box-D snoRNA. So here we're looking at all these box-D snoRNAs collapsed from their 5' to their 3', and the ASC here is what's known as the guide region, so it's the antisense element. So all canonical interactors would bind on these yellow highlighted regions, but what you can see here is that the non-canonical interactions overlap with other regions of the snoRNA, not just these guide elements. And in fact, some of these interactions don't involve at all the guide sequence of the snoRNAs. Gabriel also considered high-throughput RNA-RNA interaction data sets. Paris, Liger-Seq, and Splash. She de novo analyzed these. So all of these detect in a high-throughput manner both inter and intramolecular interactions. They involve cross-linking using psoralen and degradation of the flanking sequences using nucleases. Then ligation of the interacting RNAs, decross-linking, library preparation, and sequencing, to figure out which RNAs were originally interacting in cells. So she de novo analyzed these data sets, so more than 500 million chimeric interactions, and of those, approximately 300,000 involved a snoRNA interacting with another RNA. So she further analyzed these and compared them to the other data sets of interactions for snoRNAs. And what she found here, she was looking at the distribution of the biotypes of the interactors of snoRNAs. So for the non-canonical, or for the canonical, the known canonical data set, these all involve interactions with ribosomal RNA and snRNA, as is well characterized. For the literature data set, almost all of these involved interactions with messenger RNAs. But for the high throughput RNA-RNA interaction data sets, so snoRNAs were found to interact with many different RNAs of different biotypes, many messenger RNAs, which fits well with what has been found for the small number of examples heavily validated in the literature. But there are also snoRNAs interacting with pseudogenes, long non-coding RNAs, other snoRNAs, as well as tRNAs, snRNAs, and ribosomal RNAs. This paints a very different portrait of the snoRNA interactome. And we wanted, being a bioinformatics group, we wanted to investigate whether we could study further these interactions, obtain more information about them, and whether we could create a predictor that could generalize this information to be able to predict transcriptome wide and not be limited to the cell types that were considered and the depth of sequencing that was obtained for these interactions or for these data sets. So Gabrielle created a predictor called SNOGLOBE, and she considered for the prediction the window of interaction in the snoRNA, the window of interaction, so the sequence of the interacting window in the target, the relative position of the interacting window in the snoRNA, the position in the target, and the biotype of the target, so whether it's snRNA or messenger RNA or tRNA, for example. She investigated many different machine learning algorithms. Those that performed best were gradient boosting classifiers, which are a tree-based algorithm. So that's what she further used for the rest of the project. And then, so when we create a bioinformatics predictor, we need to have examples that we're going to present to the predictor from which the predictor will learn. So in our case, we needed positive examples of interactions between snoRNAs and other RNAs, as well as negative examples. So the positive examples I've already described to you, the known canonical, known non-canonical from the literature, and the high throughput RNA interactions from the Paris-Liger-Seq and SPLASH data sets. For the negative examples that we need to present to the predictor, this is always a problem in bioinformatics because not very many people publish papers describing non-interacting RNAs. So we created our own negative examples. We created randomly or random negative windows. So these are pairs of sequence randomly chosen from all human snoRNAs and a sequence randomly chosen from the entire human transcriptome. And we also created what we call the matched negative set. So for every positive example that we have in our sets, we created 10 matched negative interactions. One of them involved the exact position in the interaction in the snoRNA and a random position in the target. And conversely, we also have one example where we have the exact position interacting in the target, but a random position in the interacting snoRNA. And then we have 8 randomly chosen pairs of random position in the snoRNA and a random position in the target. And now we thought that it was important to have this kind of negatives as well, because we think it's harder to distinguish between these matched negatives and the positives compared to the random examples. So all of these data sets were split into non-redundant tuning, training, and testing sets. And we did this in a very stringent way to make sure that there was really no redundancy between these sets. So for example, we don't train on an example and test on it as well, but we also used RFAM clans to split the snoRNAs in these sets, so there's really not any redundancy between the sets. And so one thing that we wanted to ask by creating this predictor is we're very interested in non-canonical interactions, and we really want to be able to predict that there are few that are available in the literature, and we really want to be able to predict that transcriptome wide. So we decided to keep all the non-canonical interactions from the literature for the test set, to investigate whether we can only train on canonical interactions and high throughput RNA interactions, and then still be good at predicting these non-canonical interactions. So we created our predictor, and then we compared its performance to the performance of other similar predictors. So the closest predictors that we could compare SNOGLOBE to are Plexi and SNOSCAN, which are predictors of mostly of canonical interactions of boxity snoRNAs. So it might be a bit unfair to compare them to SNOGLOBE, which is trained and really built to also be able to predict non-canonical interactions. But that was what is currently available in the literature to make such predictions. We also compared the predictions of SNOGLOBE to the predictions of general RNA interaction predictors, RNA-Plex, InterRNA, RiseRch2, and RNA-Up. So here we're looking at raw curves and precision recall curves. In the case of raw curves, we're comparing true positive rates and false positive rates. So we're hoping that the predictor will predict with a higher score all true positives before starting to predict false positives. So we're hoping for a curve that's going straight up all the way to the left, top left corner. In the case of precision recall curves, we want high precision and high recall. So we want the highest, we want the curve to go to the top right corner. And then so what you can see that SNOGLOBE is the predictor that performs best in both cases. We looked at breakdown of the different interactions in the test set. So here we're looking at the predictions on the test set for each of the different predictors that we considered. We find that for almost all negative examples of the test set, all predictors predict them as negative, so that's good. SnoGlobe predicts many of the positive examples from the high throughput RNA-RNA interaction data set and from the known non-canonical set as positives, whereas the other predictors don't manage to identify them. And then SnoGlobe does well on many other positive examples that the other predictors also are good at identifying. So if we look in overall at all the positives in the test set, SnoGlobe identifies pretty much all known canonical interactions, and the other predictors do well on canonical interactions, but SnoGlobe identifies almost all known non-canonical interactions from the literature, whereas the other predictors are not very good for those, and it also does well for the high throughput RNA-RNA interactions. So at this point, we were ready to use the predictor and apply it transcriptome-wide for the entire human transcriptome. So we considered all predicted interactions between all human snoRNAs and the entire human transcriptome. Here we're looking at the positions in all human snoRNAs that are involved in interactions with the human transcriptome as predicted by SnoGlobe, and we're considering snoRNAs from their 5' to their 3' here in this left panel. What you can see in color here is in purple the box D and the box D' regions, as well as in green the box C and the box C' regions. So they don't always exactly overlap. That's why you see smears here, but generally the guide regions that are involved in canonical interactions will be immediately upstream of the box D and D'. So they should be in this region here and in this region here. And you can see that a lot of those predicted interactions will involve the guide regions, but there are other predicted interactions, quite a few that don't involve these guide regions, which fits well with what was seen for the non-canonical interactions in the literature. If we look at which regions of the transcriptome are predicted to be targeted by boxy snoRNAs, here we're looking at the entire human transcriptome in terms of 13 nucleotide windows, because that's the size of sequences that the predictor considers. And so the great majority of the transcriptome consists of intronic sequences, so 92% or 93%, and 7% are exonic sequences, and very few of these windows will overlap exon-intron junctions. But if we look at the predicted regions, or the regions that are predicted to be targeted by boxy snoRNAs, we see that exonic sequences are significantly enriched. So there's twice as many hits on exonic sequences as what you would expect randomly. And much more so, there's a huge enrichment for binding of snoRNAs on intron-exon or exon-intron junctions. So some snoRNAs seem to have a strong preference to binding on splice junctions. And then if we look specifically at exonic sequences, there's an enrichment for 5' binding on 5' UTRs. And in particular, some snoRNAs seem to have most of their predicted interactions with 5' UTRs. These might be involved in the regulation of translation, but we haven't, this is just speculation. We haven't started investigating that yet. I don't know why it doesn't change like, okay. So we investigated more specifically some snoRNAs and that have specific or interesting positional enrichment for their targets. So here we're looking at a collapsing of all exonic sequences, as well as 100 nucleotides upstream and 100 nucleotides downstream of all human exons. So this snoRNA, for example, has a preference for exonic sequences, but much more so for this would be 3' splice sites. This snoRNA has a preference for 5' splice sites, and this snoRNA would have a preference for polyprotein tracts and branch points. So we characterize in more detail some specific snoRNAs and their targets. I don't really have time to talk in more detail about those. I'll just mention one of them, so SNORD126. It's an orphan snoRNA, which is the term used to characterize snoRNAs that are known or not known to be involved in canonical interactions with ribosomal RNA and snRNA. And we investigated it in a collaboration with Sherif Abouelada's group, and we obtained interesting observations. So here we're looking at the snoRNA sequence from its 5' to its 3', and these are the regions that were predicted by SNOGLOBE to be involved in the interactions transcriptome-wide. So the region that is predicted most to be involved in these interactions is between the box C' and the box D', which is not a guide region. And if we look at this region in the predicted folded structure of SNORD126, it corresponds to this loop here. So this is really not a canonical interacting region for a snoRNA. And if we look at its predicted targets transcriptome-wide, we see an enrichment for exonic sequences, but also an enrichment for binding 80 nucleotides upstream of exons. And amongst the messenger RNAs targeted by, or predicted to be targeted by SNORD126, we see an enrichment for cell junction, cell projection, and plasma membrane-bounded cell projection messenger RNAs. And so there's an enrichment for binding to exonic sequences, but also binding on splice junctions for that snoRNA. So in collaboration, as I said, with Sherif Abouelada's group, we depleted SNORD126 and investigated whether the predicted targets of SNORD126 would be affected by the depletion. So here we're looking at changes in the abundance of messenger RNAs. We see many messenger RNAs go down in abundance following the depletion of SNORD126, and those that are colored in blue, green, or red were predicted by these predictors to be targeted by SNORD126. And in general, we find that SNOGLOBE, Plexi, and SNOSCAN in this case, and particularly SNOSCAN, do well in identifying or predicting targets that were affected by the depletion of SNORD126. And SNOGLOBE does best to identify RNAs that are differentially spliced following the depletion of SNORD126. And when we compare these predictions from the three snoRNA specific interactor predictors, they're much more useful than general RNA interaction predictors, which basically predict that SNORD126 would bind to every messenger RNA in the cell. So that really doesn't narrow it down if you're interested in validating this experimentally. So overall, we find that boxy-D snoRNAs interact with diverse RNA types, and that the interactions of boxy-D snoRNAs don't only involve canonical guide regions, so we should also investigate these other types of interactions. SNOGLOBE outperforms all currently available tools, and when we investigate the transcriptome-wide predictions of SNOGLOBE, we can identify regulatory modules. I touched on this a little bit, but we're currently going much further in these analyses and depleting multiple snoRNAs of interest to investigate this further. We have a script that's available to run remotely to predict SNOGLOBE predictions, but we're also creating a web server that will be easier to use. You could just paste your sequences of interest. For our future studies, we're as I said, validating further or other snoRNAs by knocking down and sequencing. We're interested in investigating the determinants of binding between snoRNAs and their targets. We want to characterize snoRNA interaction networks, including which RNA binding proteins might be interacting with the snoRNAs to carry out these non-canonical functions. We want to investigate whether snow globe functions well in other organisms, and as I said, we're creating a web server. And I'd like to finish by thanking Gabriel Deschamps-Francœur, who did most of the work here and here. This is a very recent photo. This is one from March of this year. I like this photo because we like snow in all its shapes and sizes. I'd like to thank the funding agencies and you for your attention, and I'll be happy to answer your questions.
So thanks a lot, Michelle. This is really very interesting, and I hope that we're going to get a lot of questions. We have a kind of 3 questions for now from Fabrice Leclerc, so we're going to try to go through them. The first one is actually very interesting. So Fabrice asking is really why there is a redundant snoRNA. In a sense, he's asking if snoRNA's modification site targeted by more than one snoRNA are more critical for function, or that the mean of rate of modification lower for the corresponding snoRNA, snoRNA are actually acting at a different level. That's why you need the redundancy.
So snoRNAs exist in multiple copies because they seem to be good substrates for retrotransposition and also recombination. So these are 2 mechanisms that have been used by multiple snoRNAs to copy themselves in multiple positions in the genome. Box-CD snoRNAs have a preference to copy themselves in the same, so I don't think I mentioned this, but in mammals, many snoRNAs are encoded in longer genes referred to as their host genes, and they can end up copying themselves in multiple introns of the same host gene. Box-HEC snoRNAs have a stronger relationship with retrotransposons, and they can copy themselves at any position in the genome. And we haven't seen, so there are some families of snoRNAs that have multiple copies and are predicted to all modify the same position in ribosomal RNA, but there are other, you could call them families of snoRNAs, but that in human there's only one member of this family, and they target very important positions in ribosomal RNA. So there doesn't seem to be a relationship between the number of copies and the importance of the modified site in ribosomal RNA. But we do think that if you have multiple copies, this might allow you to modulate the abundance of the RNA that's modifying a specific position. So across different tissues or in different conditions, maybe you could end up changing the abundance of the whole family and affecting a specific position in ribosomal RNA.
Okay, so we have several questions, so I'm going to try to pick up 2 for the rest of you who your question is not picked up. Michelle will answer you during the webinar or just after. So we have one here is that from Charles Bonader, and he's asking, did you check if the non-canonical targets you identified are post-transcriptionally modified? Is it possible that the regulation occurs through structural changes in the target induced by this RNA-RNA interaction? I think those are 2 separate questions. The one is it actually modifying non-canonical modification or really independent? And the other one is that is an interaction could induce structural changes in the RNA.
So there is overlap between our predictions, well, and the non-canonical or like, well, I don't. So there aren't very many validated non-canonical interactions in the literature, but if we look at the high throughput detected RNA-RNA interactions involving snoRNAs, there's overlap with some positions that are known to be modified or methylated in the case of box-CD snoRNAs, but the overlap is small. But at the same time, what we think is happening is that the depth of sequencing to detect the modified position is not great enough to be looking at the whole space yet. So we think that we're looking at sub-regions of the entire interaction space for snoRNAs and modifications, which would explain why the overlap is very small, but so this remains to be seen. And we haven't started investigating yet whether this structures are involved in these modifications and in these interactions, but we're definitely very interested in that question.
Okay, so one last question before, and the rest, Michel, please do answer them in the Q&A. That's from Bruno Charpentier, and the question is, any idea on the protein composition of the RNP for the non-canonical targeting? Is a snoRNP actually changing? Very interesting question.
Yeah, very interesting question. So the canonical interactors, protein interactors of snoRNAs are well known, but other groups have shown that in the case of snoRNAs involved in, for example, splicing, the Boxed snoRNAs interact with some splicing factors. In the case of some of other levels of regulation of gene expression by Boxed snoRNAs, there is known interactors, different known protein interactors that would be involved in the function. But we really don't. I think we're scratching the surface, and we're very interested in going further with this. In the case of our snow globe predictions, we started investigating whether there's overlap between binding sites or RNA interactions for specific snoRNAs and specific RBPs using eClip data sets from ENCODE. And we do see strong enrichments for overlap between these two data sets. But once again, we want to investigate this in much more depth.
Okay, so unfortunately, we don't have time. There's many more questions that I'm sure Michelle is going to go in the Q&A and answer them for you. But we're going to have to, for the interest of time, we have to move on. Thanks again, Michelle. Now we're going to move on to the next presentation. So our next presenter is Peter Unrau from the University of Simon Fraser University. It's actually my distinct pleasure to introduce Peter. I had the pleasure to introduce him a couple of times before, and always is a challenge because every time I try to present his CV, there is many things I don't understand, despite my kind of capacity to do that. So Peter finished his BSc in Math and Physics from McMaster University in Ontario, Canada in 1992, and he received his PhD in Theoretical Physics on hadronic spin structure from MIT, and do not ask me what that is. I will leave that to Peter to answer in your Q&A. Under the supervision of Jiang Dong in 1996, and he followed that by a complete change, I would say, in his kind of education, going to do a postdoctoral fellowship in RNA biochemistry on the origin of life. So clearly, he does not choose his topic lightly, and he finished that in 2000. He's currently a full professor in molecular biology and biochemistry at Simon Fraser University in British Columbia. He's also acting CEO of Aptamer Innovation at Burnaby. His lab focuses on RNA and its role in early evolution and modern life. He's interested in understanding and demonstrating how RNA could have started life on Earth, developing RNA tools to inform of aptamer, a new RNA amplification technology and sequencing technology. And if that is not enough for you, Peter also interested in modern RNA biology and has several contributions understanding RNA processing and contribution to cancer. His papers are published in very good journals. I don't need to cite them all. So with that, I will let Peter to present, and you're invited to ask all questions from why we're here on Earth to how RNA works. From that, Peter, take it from there.
Thank you very much, Sharif. Everybody can hear me? Yeah, so I guess for this audience, I think it's quite clear that RNA plays a key role in many diseases. And from the RNA world perspective, I think it's pretty clear why. The RNA world hypothesizes that life started with RNA and steadily embellished function by adding protein through the ribosome and translation, and embellished further the ability to store information in DNA. But it's thought that life started with RNA and catalytic reactions involving RNA, and that RNA is really a perfect tool now within modern cells to just regulate huge amounts of function. And I've just pretty much arbitrarily listed some kinds of RNA function over here that are regulated by RNA. So I got into this field of RNA research by being interested in how you set up the first self-replicating systems at the origin of life. And this is just a slide from NASA where they're showing a robot gathering parts off of a shelf, passing them to another identical robot to assemble a robot that eventually will be able to take parts off of the shelf. And that, roughly speaking, is what life looks like. There's functional elements that have to be organized so that they can replicate at a multiple sets of scales. So I've studied this problem using in vitro selection. This is a way of efficiently screening through very large populations of molecules where you throw out the inactive ones, keep the active ones, and amplify and enrich, mutate perhaps, and reselect. And as I've gone through this process of trying to discover the functionalities of RNA from the perspective of origin of life, I've had to design more and more complicated selective protocols. So when I was in David Bartel's lab, I was interested in glycosidic bond forming reactions. And so this is an essential element of making nucleic acids, where you make PRPP is converted together with a base into a nucleotide, which is eventually converted into a triphosphate. And I showed that RNA could do that reaction, but the tools I needed didn't exist at the start of my project, and led to some interesting research tools. So while I was a postdoc, we figured out how to adenylate DNA oligos. And in this way, I could ligate DNA adapters onto the end of my in vitro selection pools. And then when I reverse transcribe these pools, I could actually mutate these pools all the way down to the very 3' end of the pool, and then this way sequence RNA molecules that had functionality all the way down to their 3' end. And this was used by Nelson Lau to clone and sequence the first large set of microRNAs before there was high throughput sequencing. And you can still buy these kinds of adenylated oligos from IDT. So recently, Razvan in the lab has made an RNA polymerase that has clamping behavior, and this is shown schematically here. It has a oligo that it extends, but it uses this oligo to find a promoter and then extend it after it's clamped onto the template. And this is very much analogous to the way that protein enzymes find a promoter and extend it in an elongation mechanism, let's say in bacteria where you have sigma factors. So to go further than this, I'm just going to have to continually develop better tools. And one of the tools that I'm going to talk to today about is RNA Mango, which is a high affinity fluorogenic aptamer. All right, so this is what it looks like when you have the fluorogen mixed with an RNA Mango aptamer, and we have 3 different color classes here: teal1, YO3, and teal3. This work was largely done by Lena in the lab, Sunny, Sham, and Amir, who together with myself have founded a company called Aptamer Innovations for full disclosure. These aptamers are just very high affinity, high brightness, they're small, biologically compatible, and they have a range of interesting chemical and biophysical properties that I'm now going to spend the rest of my time discussing with you guys. Okay, so our work has been published on the left here, and it's led to utilization by research groups, largely for transcriptional reporters. So these Mango aptamers seem to be amenable for transcriptional reporters, and that's what a lot of people have used them for. That's this middle set. They've also used them for reporting on microRNA populations, but people have done some weird and interesting things, which I find interesting. They built large nano scaffolds and decorated them with mango aptamers. They pulled on them. There's just a lot of interesting utilization for these kinds, these and other from other groups, types of fluorogenic aptamers. Okay, so mango aptamers specifically, we have four main types, and I'm going to largely in this talk focus on mango 2 and mango 3. Together with Adrian Perry-Damari's lab, we have crystal structures of mango 2, which is here, mango 3 on the right. And they have interesting differences. Mango 2 is floppy. It has a fluorogenic module that's built out of a G quadruplex that binds tightly to its dye, but that can flop around in space relative to a stem loop, which has a little adapter motif that feeds it into this quadruplex core. So this can go off into biology very easily, and this can be the fluorogenic reporter. In contrast, mango 3 fits solidly onto its helical stem via a triplet of RNA here that docks onto the stem and then into the quadruplex that binds the fluorogenic molecule. So this is mango 3 is a rigid fluorogenic aptamer. Mango 2 is a floppy fluorogenic aptamer. Okay, so if you want to characterize these things, this is the TL1 biotin ligand that we like to use because it enables fluorescence and pull down assays. This is that fluorophore in a tube. If you add an aptamer, that's what you're going to see. And that's the fluorescent enhancement, which is on orders of thousands or up to about 4,000-fold enhancement from the background fluorescence of this molecule, being brighter than ultimately than GFP. So that's fluorescent enhancement. They also have very high binding affinity, which is in the low nanomolar for mango 2. And it's this property of being in the upper left of this quadrant that makes these aptamers so interesting to us. One key reason for that is the fluorescence efficiency, which is analogous to the enzymatic efficiency for an enzyme. If you just take the fluorescent enhancement and divide it by the KD, that's a characteristic number. The larger that number is, the more fluorescently efficient you're going to be. And that corresponds really to how much contrast you can expect to have inside a cell. There's lots of other factors that will take away from that contrast, but that raw number is an interesting metric, and mango aptamers do quite well compared to other fluorogenic aptamers. All right, so I'm just going to talk through 4 different properties of these mango aptamers. First, I've mentioned is that they're high affinity, but that just enables rapid RNA-protein complex purification from cell extracts. And we've done this in a number of different contexts, both bacterial and eukaryotic cells. So in the lab, we've been studying the 6S regulatory RNA. It's an RNA in bacteria that throttles transcription of housekeeping transcriptional processes. So the sigma 70 promoter bound to holoenzyme. Normally, you think of it binding a DNA promoter. It then transcribes it. It kicks out the sigma 70, so it can be recycled. It kicks out an RNA transcript and ultimately a piece of DNA. This core enzyme then rebinds a sigma 70 factor. But if there's lots of 6S RNA around, it's going to bind upwards in this pathway. And depending on the rate of release of the 6S RNA, it's going to actually inhibit this bottom transcriptional process. And so in stationary phase cells, you actually have the majority of sigma 70 holoenzyme in this state, as Wasserman and others have shown. And when you go into rich media, nucleotide triphosphate levels, which are required for actually producing a small pRNA that triggers the rearrangement of the 6S RNA and its release, triggers its rapid releasing the sigma 70, the core enzyme, so that housekeeping transcription can rapidly resume. So it's a throttle. So Sham in the lab demonstrated that you could just pull out of bacteria this complex of the 6S RNA and RNA polymerase. He could bind it to beads using this substrate I just showed you, the TL1 biotin. In fact, he was using deciobiotin. After washing and eluting with biotin, he could then pass that through a size exclusion column and get very pure enzyme comparable to multi-step purification of commercial RNA polymerase enzyme. Recently, Chris in the lab has also mango tagged the 6S RNA. He's also done pull downs, but what he was interested in is the correlation between the pRNA production and the maturation of the 6S RNA. So at the 5' end, you can have different RNases cleaving the 6S RNA. And he could observe the pRNA length and the maturation at the 5' end of the 6S RNA simultaneously as a function of growth by pulling out the 6S pRNA complex. This pRNA is what actually triggered the rearrangement, so it released. and then just adding RT enzyme just to fill in the 6S RNA using the pRNA as a primer. And then this way he could observe the length of the 6S RNA, which is not fully matured at early time points. And then its maturation as you undergo outgrowth, and the size of the pRNA when he hydrolyzed off all of the pRNA and then measured that indirectly through the length of the cDNA that he produced. More interestingly, he observed that in stationary phase, up to 30% of these pRNAs are in fact not triphosphorylated at their 5' end, but they're actually capped with nicotinamide. And this really suggests, and I won't have time to fully explain this, but really suggests that in stationary phase, at least some of the cells have very little ATP in them. And this follows from the fact that nicotinamide levels are competing for initiation for pRNA synthesis, as demonstrated by this data. Switching to yeast and a collaboration with yeast experts, we have pulled down different parts of the spliceosome using a Mango tag, not interfering with the spliceosomal function, and pulling down proteins that are associated with each of the spliceosomal RNA-protein complexes. All right, so the fluorogenic properties are also interesting, and you get those simultaneously with the single tag of inserting into your RNA of interest. And so I'm going to talk about fixed and live cell imaging. So this is Chris's work again. He's just demonstrating that he can see the 6S RNA in stationary cells using the Mango tag that he inserted into the loop of the 6S RNA hairpin using Mango 3. This is work from David Rauda's lab in collaboration with my lab, where they did structured illumination using the Mango 2 aptamer in fixed conditions. Interestingly, this is, these aptamers are still well folded in 4% formaldehyde. I find this data particularly exciting. Normally, photo bleaching is not something one gets excited by. This is demonstrating photo bleaching of a Mango aptamer in 5 seconds. What's interesting is that after 5 seconds of having the laser off, and using liquid media containing the fluorophore that binds to our aptamers, we've completely recycled that fluorescence cycle. And that can be done up to 700 times before we got bored. Right, so we can continuously image these systems by fluorophore recycling. This is again a Mango 2 aptamer, which is the floppy aptamer, which binds to a very high affinity, compared with the MS2 protein imaging system. Here we just have the MS2 system, if you're not aware, is just a Mango tag that binds tightly to our protein that's derivatized with a fluorescent protein, allowing it to be imaged. And as expected from that fluorescent efficiency plot that I just showed you, we see much better contrast here, of course, in the individual molecule of RNA than we do with the gold standard MS2 system. We've been developing multi-channel imaging capabilities. So Mango is normally used in the green channel, compatible with let's say GFP color filters. Peach is much redder, and this system we just developed recently, where we also have a floppy system. We haven't got the crystal structures yet, but it looks very much like Mango 1 and Mango 2, which have this, a tetraloop-like adapter on it. But it binds a very similar fluorophore, but with a completely different color than the Mango 1 and 2 and 3 systems. And that's demonstrated here, where we're just displaying binding orthogonality of the Peach system and the Mango 3 aptamer for RNA concentrations ranging from 0 to 50 nanomolar. Here we're increasing the Peach concentration from 0 to 50 as we decrease the Mango 3 concentration from 50 to 0. Here we're simultaneously increasing both from 0 to 50 and reading out these concentrations just from a perimeter. The last thing I'll talk to you about are RNA sensors and reporters. And I think there's a lot of mileage in this area that my lab is currently concentrating on closely. So I think it's very interesting to be able to have FRET sensors inside cells that can read out some kind of RNA state. And This is just really a demonstration that we can do this in an interesting way using what we call oriented fret. Normally you think of fret as just a distance-dependent phenomenon, that the fret efficiency scales as 1 over r to the 6th, with some characteristic distance that is characterized by the fret pair that you're using and the solvent that you're in. But it depends on kappa squared, which is dependent on the relative orientation of the acceptor and donor dipoles. So in this data, what we're doing is we're putting the broccoli aptamer, which is rigid, and we're coupling it via fret to the Mango 3 aptamer at the end of a helix. And so because these two fluorogenic aptamers have helical elements, and because the acceptor and dipole, acceptor and donor dipoles are roughly speaking perpendicular to that helix, occasionally as you extend the helix, you'll go through periods where the two dipoles are parallel, and therefore the fret will be maximal, or perpendicular, where kappa squared will be really small and minimal. And then as you rotate around this helix, you get further away, true, but you now are anti-parallel and therefore maximal again, perpendicular again and therefore minimal again, and then parallel again as you proceed outwards. And so this has been strongly enabling. So we decided to study this using the cobalt riboswitch. This is a 4-helix structure that, based on its crystal form, stacks P2 and P4 in a collinear fashion when you add cobalt or nickel. Here we're using cobalt. But we inserted a mango aptamer and a broccoli aptamer into these two arms, and just observe the fret signal that we see as a function of cobalt. We had a very nice titration. And then as we change the position in these helical elements, we again see these very nice oscillations that we have yet to fully model, reflective of the exact angle between the P2 and the P4 helices in the rigid structure. Here's the 0 cobalt data, which is just showing this minimal fret signal for multiple constructs. I think there's at least 20 constructs that we had here, but very, very easy experiments to measure these fret values. Okay, so I'd like to just conclude by mentioning all the collaborators on this work. My current lab on the left. I also have some very talented pumpkin carvers. That's largely Ikra, actually. But I'd like to mention the funding sources and the fact that our department is currently searching for an assistant professor position in nucleic acid biochemistry quite generally. So if you're interested in that opportunity, please just contact my chair, Lisa Craig, at this email. And so thank you very much, and I welcome any questions.
Thanks, Peter, for all of those kind of interesting tools and ideas. I don't have yet any question. I don't see any question in the Q&A. I'm gonna, oh, there is one. That's from Charles Bonader. Is an innate immune response triggered in the presence of the synthetic mango aptamer, interferon response, etc.? Does it limit its application in human cell culture? So the idea is it's cytotoxic or inflammatory or otherwise.
Not to my knowledge, but I guess it's possible that you could get such a response. I think the trick is to insert these things into structures that you already know have an RNA stem loop helix in them. And then those seem to be very well tolerated in our experience.
But at least with your collaborator doing this in cell culture, you don't see any clear cytotoxicity, right? No, I don't think so. We see phototoxicity, but I think that's a fairly common problem that people have with imaging projects. Okay. Any other questions? So maybe I'll ask a question. So while we're waiting for more questions, because we had some experience with Mango, as you know, and one of the kind of limiting part was always, at least for pulling down or identifying them, was the dye that's needed to pull down the aptamer. Do you now have solve this supply problem to be able to make it more kind of available for the public? I mean, it's one thing to have an aptamer, but another thing to use it, right? Yeah, I mean, our laboratory decided to sell these fluorophores through a small company in the Lower Mainland called Applied Biological Materials. So they sell that hopefully for a fairly reasonable price, and it's
They sell, I think, 4 different versions of these fluorophores.
Okay, so that's interesting. I think for everyone who is interested in using Mango, there's some kind of commercial sources to do that. Any other questions?
I think there's a question about what is the RNA binding part of the aptamer?
That's in the chat. Yeah, well, good.
So for Mango 2, for example, you have this P1. They always have a closing stem, which we call the P1 stem, and after the riboswitch community. But you have a closing stem. This can feed off into your biology someplace. And then they always have some kind of adapter element. This is the GAA type adapter that feeds into a quadruplex structure that serves to bind the thiazole orange moiety here. And this is analogous. It has, this is the rigid one that I was talking about at the end of my talk. And this is the flexible one that we use a lot for imaging. And that is, interestingly, formaldehyde resistant. It's not that it doesn't react with formaldehyde, it just doesn't care that it has formaldehyde covering it. So it's
okay, so I just want to remind everyone to use the Q&A and not the chat. I'm kind of by chance looking at some. And there's one question here in the chat before we switch to the Q&A. Could this system be used from Michael Charette? Could this system be used to create probes for northern blots, such as for pre-ribosomal RNA processing northern?
Yeah, I mean, you definitely could use them as probes, I suppose. I mean, you would just have to extend the 3' or the 5' end to be the part that would hybridize. I guess you just have to decide how the cost of these things compares to, well, you have to order the oligo, which would have to be longer relative to ordering a shorter oligo that would have to have, I don't know, Psi 3 or Psi 5 attached to it or something like this, right? Or radio label. In principle, they would work. I would think you'd get more sensitivity using radioactivity, frankly, but.
Well, there is many universities who cannot use radioactivity, so that's probably why what stems from there. But the cost, I think, would be a major issue here that might be prohibiting for some. There is 2 other questions here in the Q&A. One from Jin Lee, and is the binding sensitive to oxidization as it contains G4?
I'm not sure I understood the question. Is the binding of?
Is the binding sensitive to oxidization? So I think he's saying, well, since you have the G4 quartet, you would be sensitive to oxidization or not?
Yeah, I mean, well, it's definitely sensitive to photobleaching, which is a form of oxidation, right? So when these fluorophores are oxidized, they don't bind to the aptamer anymore. Is one way of answering that question. I think you're talking about oxidation of the fluorophore.
Okay. So now another the question are coming here fast. So I'm going to try to take a few more, and then I think after that, Peter, I will have to leave it to you to answer them. But we have one here, Olivier Das, saying, amazing talk. Using RNA Mango arrays, you can do single molecule imaging. Do Mango and Peach also have single molecule sensitivity as single fluorophore? Can you do SM threat of Mango with Peach?
Yeah, that's a good question. We don't know yet, and people in the lab are currently finding out. Peach is very much analogous to Mango 1, and Mango 1 is what we found originally when we first published our work, and it's only when we reselected Mango 1 did we get Mango 2, Mango 3, and Mango 4, which have superior properties to Mango 1. So Peach 1 has probably got things wrong with it, is one way of saying it. But a variant of Peach might be quite interesting from the point of view of those kinds of experiments.
Excellent. So one last question before, and the rest, hopefully, Peter, you can just answer on later on. That's from Leslie Parent. And can you do FRAP experiments with Mango?
Well, I have to remember, I'm not a microscope expert here. I just collaborate with people who are. But the only limitation on a FRAP experiment would be the rate at which the fluorophore diffuses into the frapped area. So you might end up just measuring the rate at which the small molecule fluorophore diffuses into the frapped area. So maybe it wouldn't be ideal, I suppose.
Okay, so with that, I would like to ask to thanks Michelle and Peter for those exciting talks. And we close it here. If you have questions, you can always email Peter or Michelle or post them in the Q&A and they will continue. So thank you all for attending this kind of special edition, the Riboclub edition of the RNA Collaborative. And I would remind you again that these talks will be available on YouTube also, so you can refer your colleague or go back and see them yourself if you want to. And looking forward to see you next time in the next edition of the RNA Collaborative.